First, a little confession. I am a membrane biophysicist who sort of stumbled his way into virology. So I'm very excited to be here talking with you today, to get your feedback and to learn from you. Today I am going to be talking about an interesting tool that we have developed, these DNA-lipid conjugates, which we can use to separate virus receptor binding from membrane fusion. So as we all know, receptor binding necessarily precedes viral entry. This is true for influenza virus, which is the specific case that I'll be talking about today. But of course, it's true more broadly. For influenza, the viral hemagglutinin, or HA here, binds to a sialic acid receptor on the host cell membrane. It gets internalized by endocytosis. And inside the endosome, the pH drops, which triggers a very large conformational change in hemagglutinin, leading to fusion, whereupon the viral RNA is released and replication can begin. So our question really was, can we develop a generalized strategy to bind envelope viruses to some type of a target membrane in the absence of receptors? And the reason that we would be interested in doing that is several-fold. It would immediately enable all sorts of interesting biophysical and biochemical studies where we could look at the influence of receptor binding on fusion, where we could start to study fusion for viruses where we don't know what the receptor is, where it's difficult to isolate, and maybe some ideas in cell culture as well. So discussing this strategy is what I'm going to be talking about today. The central objectives are, first, I'm going to talk about our method using these DNA-Lipids to bind influenza virus to a target membrane in the absence of a receptor. And then I'm going to use that strategy to address a sort of biophysical question about the influence of receptor binding on the influenza virus fusion kinetics. So, and as you'll see, I'm going to be using a lot of simplified biophysical methods to assess and use this tool, but the hope is that it will be applicable more broadly as well. First, some background about these DNA-Lipids. They were developed in the Boxer lab, and they are basically, you've coupled together a short DNA oligomer to a lipid tail group by phosphoramidite coupling. This is the same reaction that's done on a DNA synthesizer, so it's very straightforward. And the nice thing about these DNA lipids is you basically put them in aqueous solution with another membrane, and they incorporate spontaneously and basically irreversibly into those membranes. You can then use, if you have complementary DNA strands on two different membranes, now you can use hybridization to bind the membranes together. And since we have control over the synthesis here, by changing the DNA sequence, we can change the binding energy, the binding orientation, who binds with whom, and who doesn't bind with whom, and so on. Now, this strategy has been very well validated in simple model membranes, and the question is, will it work in the context of the more complex membrane of the viral envelope? And so, ran an experiment to test that. This is the experimental design. We make a model membrane in a microfluidic device. That model membrane is a glass-supported bilayer, which is basically designed to be a very simplified plasma membrane mimic, but over which we can exert a lot of control. And it's made by rupturing small liposomes or small vesicles on a surface where they merge together to form a continuous fluid membrane. This membrane will then display a DNA lipid, the purple guys here, and then we can introduce fluorescently labeled influenza virus that contains the complementary DNA lipid, observe binding by fluorescence microscopy, and quantify it by image processing. And the question really is, will this process specifically work to bind the virus to the target membrane in the absence of the native receptor? And indeed, we observe that it can. What I'm showing you here is this is the number of viruses that are bound per field of view in the microscope. And what we see is that if there's complementary DNA on both the virus and the target membrane, then we get some amount of binding. If we increase the amount of DNA added to the virus, we get more binding. And in our various controls where we have non-complementary DNA or no DNA on one surface or the other, we get little background binding. So this really tells us that these DNA lipids can be used to specifically bind an enveloped virus in the absence of a receptor. So next, let me talk about using this strategy to look at a particular biophysical question about receptor binding on the fusion kinetics. And to sort of set the stage, let me give a little bit more background. So the influenza virus inside the endosome is bound to a receptor, which is a sialic acid terminated glycan. And remember that low pH is the trigger for fusion. And it actually turns out that there are several lines of evidence that suggest that multiple hemagglutinins need to engage the target membrane in order for efficient fusion to occur. So just one doesn't give you efficient fusion. And our question really is, how does this binding of the receptor influence the subsequent fusion kinetics? So clearly the trigger is not the receptor binding, right? It's pH. But you might imagine, for example, that receptor binding could cluster the hemagglutinins together, which could initiate sort of a hotspot for fusion, right? So it might speed things up. Alternatively, you could imagine that being bound to the receptors might sterically hinder the transition that you need to undergo to get fusion, in which case it could slow it down. Surprisingly, the sort of general assumption in the field has been more or less that receptor binding doesn't influence the subsequent fusion kinetics. So now we have a method to test that using our DNA lipid approach. This is the experiment that I ran, so it's very similar in many regards. First, we make a target membrane in the microfluidic device. In this case, the target membrane are these DNA-tethered vesicles, which I'll talk about here in a second. And the virus is either bound by the receptor or by the DNA lipid. We then flow in a low pH solution to mimic that pH change in the endosome, and then monitor the fusion kinetics by a lipid mixing assay, so by fluorescence microscopy. And our question really is, are the fusion kinetics where the virus is bound by the DNA lipid going to be different than when it's bound by the receptor? But first, let me talk about these target membranes. These are made by incorporating two different DNA lipids into small vesicles. So these are about 100 nanometers across. So the teal strand and the purple strand are not complementary, so they don't bind to each other. The teal strand is used to tether the vesicle to a cover slip that displays the complementary strand. And then the purple strand is used to bind the virus to the target membrane. Now, the reason that we're using these sort of funny little tethered weather balloons is because we want a membrane that's close to a glass cover slip so we can do our sensitive fluorescence microscopy, but we want it distanced enough that the interaction with the substrate doesn't dominate our subsequent fusion measurement. And I should mention, if in the case of binding to the receptors, then the purple DNA would be omitted and we would have the receptors instead. And so once the virus is bound, then we flow in this low pH solution to trigger fusion. This is an example of an individual virus fusion event where the virus is bound by these sialic acid receptors. So here on the left, you can see these are diffraction-limited spots, which are individual viruses that are fluorescently labeled. In the same field of view, we have a pH indicator membrane, which is used to sensitively monitor the pH transition at the surface. And over here, this is a video of this process taking place. So at the beginning, a low pH solution is flowed in, and over time you see that the vesicles the viruses start to blink on, and that's because there's a self-quenched dye in the viral envelope, and it dequenches upon transfer to the target membrane, so we see an increase in fluorescence. This is a trace showing that happening, so there's a pH drop, there's some waiting period, and then we see an increase in fluorescence that indicates that lipid transfer has occurred. So to characterize the kinetics for a cohort of viruses, we do this measurement for all of these individual viruses and compile it into a cumulative distribution function where we see the proportion of fusion events that have occurred as a function of this waiting time. Here are the kinetics when the virus is bound by the receptor, and our question, again, is what's going to happen when it's bound by the DNA lipid? Is it going to get faster, is it going to get slower, or is it going to stay the same? surprisingly, we found that when it's bound by our DNA lipids, within measurement error, the kinetics are basically identical. And sort of going a step further, we said, okay, well, let's increase the concentration of DNA lipid added to the viral envelope. And across a fourfold change in DNA lipid concentration, the kinetics remain essentially identical. So this we thought was really exciting because not only does it tell us interesting information about the receptor binding, but it also suggests that our DNA lipids aren't gumming up the works as we maybe could expect. To conclude, I've shown you that these DNA lipid conjugates can be used to bind a virus to target membranes in the absence of receptor, and that the lipid mixing kinetics didn't depend on binding to the receptor, which suggests that at least the rate-limiting step of the lipid transfer mechanism doesn't depend on being bound to a receptor. Finally, I just want to say, as I mentioned at the beginning, we're excited about the possibilities for these DNA lipids because we think we can start to use them to look to compare fusion between viruses where there's different receptor binding affinities or where we don't know what the receptor is or it's difficult to reconstitute, especially important for emerging envelope viruses. And we're also interested in looking at essentially identical questions that we looked at with influenza, but with other viruses where maybe we might expect more of a difference in the receptor binding. And with that, thank you to, I'm actually a postdoc in Virginia in Peter Cason's lab, and I've been doing a lot of this work, all of this work as a visiting postdoctoral scholar in Steve Boxer's lab, and members of both of their labs have been very helpful. And with that, I will take questions.
How easy it will be to play with the lipid composition on the vesicle, just to see how much the lipid influence fusion?
Yeah, this is actually a question that we're really interested in. I didn't talk about it today, but you can vary the composition not quite arbitrarily, but almost arbitrarily. So we've been looking at the influence of different components of the lipid composition, and that's sort of a story for another day. But yeah, that's one of the immediate applications of this type of a technology.
Could you say a little bit more about the practicalities, like how many of these DNAs per liposome is a good number? What's the best melting temperature? And how easy is it to modify standard oligosynthesis to include the lipid phosphoramidate?
Yeah, so that's a couple questions. In terms of the melting temperature, we've only tried DNA lipid sequences that have melting temperatures above room temperature, which is what these were. They're actually well above room temperature. We haven't pushed it down much lower than that, but it's easy enough to try. In terms of the numbers per virion, we've done some single molecule measurements sort of assessing that. The distribution is actually quite broad, more broad than you would maybe expect, say, if you were incorporating it into a simple model membrane. And it's something that we're trying to tighten up, but we haven't got there yet. But I mean, many of I guess I have some data here I can show. If you look at, for example, sort of the lowest concentration here, and you look at the number of DNA lipids per virion, the bulk of them are sort of, let's say 20 or less. And many of them have, just 1 or 2. Now, we haven't correlated this with our fusion measurement, so we don't know which portion of that population we're looking at in the fusion measurements. But that's sort of the data that we have right now. And then you had another question about, how easy is it? It's easy enough that I'm a biophysical chemist and I can do it. You can do it on the oligosynthesizer, and that reaction can very easily be done. There are commercially available cholesterol-tagged DNA lipids, if you want to call cholesterol a lipid. The difficulty there is cholesterol actually comes in and out more easily, so it doesn't stay as stably put in the membrane.
So How much do you know about what the proximity is between the interface and what the area of the interface is? And does that really constrain how many interactions you can have and therefore the dynamic range of increasing that concentration and observing differences in fusion kinetics?
Yeah, so in terms of the distance, it's a little bit, I didn't really go into this. If we jump back here to where I've designed this. What we're showing here is, so there's a 24-mer sort of sticky end, and then there's a T24 linker, poly T24 linker that basically just adds some flexibility. So a hybridized 24-mer should give you about 8 nanometers. But now you have a flexible region that allows, we wanted to incorporate that in so that whatever happens during the fusion transition isn't inhibited by the DNA duplex there. We haven't played as much with the distance between the virus and the target membrane, although that's something that we're actually working on right now. And then in terms of the interface, we know almost nothing. So these are all diffraction limited measurements. We haven't done any super resolution to try and get a sense of what the size of the interface might be.
Yeah, just because the curvature is the opposite of what you would expect and potentially could really limit the amount of interface.
Right, so you might have 20 available tethers, but you can only form 2 of them, for example. That's right.
Does your model allow you to also look at uncoding events in addition to fusion events? And can you modify the system to tag capsid proteins so you can actually look at binding of naked viruses?
So what was the first part of your question?
If this also allows you to look at uncoding of the capsid as well as fusion.
Oh, uncoding of the capsid. People have done that. Well, let me think. They've looked at pH transfer to the inside of single virions, but that is not necessarily the same as uncoding. We don't have a fluorescently tagged version of sort of the interior proteins, but that's certainly possible, and other people have done that. So it's something that we could do, we haven't done it yet. Does that address what you were asking?
Yeah, the second one was adapting it to naked viruses to look at fusion.
To naked viruses?
Yeah, see, look at binding.
Without an envelope. I'm sorry.
You can put a nucleic acid tag on the proteins of the capsid.
Yeah, that's something that we haven't really thought of doing. We've sort of mostly been focused on the envelope viruses. But in principle, it seems like that might be possible, but we haven't really considered that. That's a good idea. Thank you. Thank you.